Hi everyone, it's Morgan again. Starting off with this interactive lab that we're doing with the gummy bears. It was hard not to eat them, first of all. One of my favorite candies in the whole world. As well, just a little background on the gummy bears, which is stated in the description of the assignment. It's composed of sugar, starch, and gelatin, which gelatin is a polymer, which in turn acts like a cage in its dimensional structure. And then as well, since we're adding water and salt water to these little gummy bears, since the actual configuration of the ingredients doesn't have water, it absorbs, as you see in the gummy bears. When there is water, just pure distilled water, it'll expand. As well, with the salt water, it'll contract. This takes a while to fully be able to increase the volume and size or decrease. So that's why the time was 6 hours to 10 hours. As well, these different environments change the volume and size. There's a better reasoning in why these changed it. As well, these gummy bears were sitting in that solution. There was two types of solutions, a hypertonic and hypotonic, which hypotonic would be a water solution, pure water solution. Hypertonic would be a salt water solution. This in turn, hypotonic, which means that there's a low osmotic pressure compared to the other solution. So, which then, in talking about human cells or like blood vessels, it expands the size, as we see with the gummy bears, increase because the volume, the water is absorbed. As well, it's entering, so the water's entering the cell or the gummy bear, and it's expanding its volume and its size. Compared to the hypertonic, which has a higher osmotic pressure outside compared to inside, so which decreases its size and volume, which then in turn makes it more toxic and could potentially, it breaks down the molecules, which in a human cell, it becomes potentially shrunk, if that makes any sense. Personally, I like to compare it to us and just our anatomy and our physiology. When, or even plants, if you look at a plant that has decreased this, especially if it has a lot of salinity and it's not able to, doesn't have the barriers of a salinity protection, it becomes wilted and decomposed. But when we have a lot of water, it increases the volume, which then it bloats or fills the captivity of the the structure. So what we really want is isotonic, which then water's coming in and out the same amount. So when a hypotonic means there's more water going into the solution or into the cell or into the gummy bear. But the hypertonic, you're having water coming out of it. It's taking all the moisture out of the cell or the gummy bear, which then decreases it. I thought this was interesting. I've seen this kind of topic explained many different ways. Getting my degree at UH, we talked about more of the cells, but it's fun to make it about candy too. It's a good way to show how something increases and decreases by different osmotic pressure, as well as fluids going in and out with its different salinity. That's pretty much it for this lab. I think the biggest thing is showing how one instance something can increase and versus decrease because of its pressure and solution it's in. So thank you guys and have a good day.